Okay, so for the next several slides, I want to attempt to describe for you how synaptic interactions between the three cells within the vertical pathway of the retina, the photoreceptors, bipolar cells, and retinal ganglion cells, determines the nature of the center response. Whether a given retinal ganglion cell is on center or off center depends upon the synaptic interactions among these three cells within the vertical pathway. So as you've already learned, all of our photoreceptors, our rods and our cones, hyperpolarize in response to light stimulation. And as you've already seen, some retinal ganglion cells depolarize and increase their firing in response to light stimulation of the center zone, whereas other retinal ganglion cells hyperpolarize and decrease their firing in response to light stimulation of their center zone. The explanation for this difference comes from the nature of the response of bipolar cells to glutamate that's released from their presynaptic photoreceptors. And in addition, the bipolar cells release glutamate onto the retinal ganglion cells. So at the synapse between photoreceptor and bipolar cell, we can have one of two things happening. Some bipolar cells are normally excited by glutamate, and some bipolar cells are actually inhibited by glutamate. But the synapses between all bipolar cells and all ganglion cells involves glutamate, and glutamate always has an excitatory effect on retinal ganglion cells. So let's take a closer look at how bipolar cells respond to glutamate and to light stimulation of their presynaptic photoreceptors. So we can distinguish two types of bipolar cells. On-center bipolar cells provide synaptic excitation to on-center retinal ganglion cells, and off-center bipolar cells provide synaptic excitation to off-center retinal ganglion cells. All bipolar cells receive glutamate synaptic input from their presynaptic photoreceptors, but on-center bipolar cells hyperpolarize in response to glutamate. In other words, on-center bipolar cells are inhibited by glutamate. Now, when we studied glutamate and glutamate receptors previously, you learned that glutamate has an excitatory effect on postsynaptic cells. This is true for all synapses within the nervous system, except for synapses between photoreceptors and on-center bipolar cells, where the nature of the synaptic response to glutamate is inhibitory. So while on-center bipolar cells are inhibited by glutamate, off-center bipolar cells are excited and depolarize in response to glutamate stimulation. And then finally, our bipolar cells, which provide synaptic input to our retinal ganglion cells, always have an excitatory effect on retinal ganglion cells. So all retinal ganglion cells depolarize in response to glutamate stimulation. Okay, so let's take a look at how, first, off-center bipolar cells respond to light stimulation of their presynaptic photoreceptor, and how the off-center bipolar cells are affected by glutamate. So our off-center bipolar cells are our bipolar cells that have a normal response to glutamate. So off-center bipolar cells contain either AMPA or kainate type ionotropic glutamate receptors. These are glutamate receptors that form a cation channel, so glutamate stimulation of the glutamate receptors on off-center bipolar cells causes depolarization. Okay, so now let's consider how light stimulation would affect the presynaptic photoreceptor and the off-center bipolar cell. So you'll recall that all photoreceptors, all rods, and all cones hyperpolarize in response to light stimulation, and that's illustrated in the upper right-hand portion of the slide. So if this presynaptic photoreceptor hyperpolarizes in response to light stimulation, it's going to release less glutamate onto its postsynaptic off-center bipolar cell. Now, the off-center bipolar cell is normally going to be depolarized by glutamate because it contains conventional ionotropic glutamate receptors. So in the dark, in the absence of any light stimulation of the photoreceptor, the off-center bipolar cell is receiving a constant glutamate input that's keeping the off-center bipolar cell depolarized in the absence of light stimulation. If we stimulate the photoreceptor with light to cause a hyperpolarization, the off-center bipolar cell is going to receive a smaller glutamate stimulus, so its membrane potential will actually hyperpolarize. It loses some of the excitation that it normally receives in the dark. So the off-center bipolar cell will then hyperpolarize in response to light stimulation. And as the off-center bipolar cell hyperpolarizes, it's going to release less glutamate onto its postsynaptic retinal ganglion cell. So because the postsynaptic off-center retinal ganglion cell gets less excitation when the photoreceptors in the center of its receptive field are stimulated with light, it also hyperpolarizes and decreases its firing rate. So this is the nature of these synaptic interactions within the vertical pathway, where all of the synapses are glutamate and all of the synapses have an excitatory effect on postsynaptic cell. This determines why the off-center retinal ganglion cell hyperpolarizes in response to light stimulation of photoreceptors in the center zone of its receptive field, and why the off-center retinal ganglion cell decreases its firing rate when photoreceptors in the center of the receptive field are stimulated with light. All right, now let's take a look at how on-center bipolar cells respond to light stimulation of photoreceptors that are presynaptic to the bipolar cell. So on-center bipolar cells are normally inhibited by glutamate. This is a unique response to glutamate that we find only at synapses between photoreceptors and on-center bipolar cells in the retina. And this unique response to glutamate depends upon a metabotropic type glutamate receptor that's uniquely expressed in on-center bipolar cells. Now, I've indicated that this is metabotropic glutamate receptor subtype 6. There are actually at least 7 subtypes of metabotropic glutamate receptors. This specific metabotropic glutamate receptor is expressed only by on-center bipolar cells within the retina. Now, while I don't need you necessarily to remember that it's metabotropic glutamate receptor subtype number 6 that's involved in this response, I do need you to appreciate that because this metabotropic glutamate receptor has a hyperpolarizing and influencing and inhibitory influence on the on-center bipolar cell, when the on-center bipolar cell is stimulated with glutamate, it will be hyperpolarized, unlike all of the other cells in the nervous system that are depolarized by glutamate stimulation. So if the photoreceptor, like all rod and cone photoreceptors, hyperpolarizes in response to light, it's going to release less glutamate onto its postsynaptic bipolar cell. And if that postsynaptic bipolar cell contains only metabotropic glutamate receptor subtype 6, it is normally going to be inhibited by glutamate, hyperpolarized by glutamate. So in the dark, when the photoreceptor is as depolarized as it ever is and is releasing glutamate at a high rate, the on-center bipolar cell is kept hyperpolarized. And if the photoreceptor that's presynaptic is stimulated with light, it hyperpolarizes and stops releasing glutamate onto the postsynaptic on-center bipolar cell. So the postsynaptic on-center bipolar cell is released from inhibition when its presynaptic photoreceptor is stimulated by light. And because it's released from inhibition, it's able to depolarize in response to light stimulation of its presynaptic photoreceptor. So if the on-center bipolar cell depolarizes when its presynaptic photoreceptor is stimulated with light, it's going to increase glutamate release onto its postsynaptic on-center retinal ganglion cell. And because the retinal ganglion cells all contain conventional ionotropic glutamate receptors, kainate or AMPA type glutamate receptors, when the on-center bipolar cell depolarizes and increases its glutamate release, it's going to depolarize the on-center retinal ganglion cell to cause an increase in firing in the on-center retinal ganglion cell. Okay, so that's how the synaptic interactions between photoreceptors, bipolar cells, and retinal ganglion cells determines the nature of the center response of retinal ganglion cells. On-center retinal ganglion cells receive their synaptic input from on-center bipolar cells that are normally inhibited by glutamate, but which depolarize when their presynaptic photoreceptors are hyperpolarized by light. And for off-center retinal ganglion cells, they also receive synaptic input from off-center bipolar cells, but because off-center bipolar cells lose excitation when their presynaptic photoreceptors are stimulated with light, the bipolar cell and the off-center retinal ganglion cell both also hyperpolarize in response to light stimulation, and that's how we can get an inhibitory or an off response to stimulation of the center zone of their receptive fields. Okay, so the next thing that we need to consider is why the surround zone has an antagonistic response. So in considering the nature of the response of the surround zone, it's functionally antagonistic. So light stimulation of the surround zone always produces the opposite type of response compared to the center. It doesn't matter if we're talking about an on-center retinal ganglion cell or an off-center retinal ganglion cell. The nature of the surround response is always the opposite of the center response. And this antagonism between center and surround of the receptive field is produced by a specific cell type that's functionally antagonistic or inhibitory, and this is a horizontal cell. So as the illustration on the right of the slide shows, horizontal cells essentially link photoreceptors in the surround zone to photoreceptors in the center zone. And this inhibitory linkage between photoreceptors in the surround and the center is what creates the functional antagonism between center and surround zones of the receptive fields of our retinal ganglion cells.